We'll move on now to the short talks. Our first is from Paulina Villaseñor. She's a master's student in the Brain Connectivity Lab at the Instituto de Neurobiología de la UNAM, and will talk to us today about microstructural analysis of the cortex using diffusion-MRI and histological validation in an animal model of cortical dysplasia. Looking forward to your talk, Paulina. Take it away.
Thank you. I'm going to share my screen. Okay. Can you see my slides?
Yes. Perfect.
Okay. Okay, well, thank you very much for the introduction. My name is Paulina Villaseñor, and today I'm going to talk about the microstructural changes in the cortex using diffusion-MRI and histology in our animal model cortical dysplasia. So I would like to start with a research question, which is if can diffusion-MRI detect the microstructural changes in focal cortical dysplasia? So far, we know that FCD is a malformation of cortical development that causes drug-resistant focal epilepsy and is characterized by cortical eye disruption and abnormal neurons, as I show you here in this representation. In your left-hand side, you see a normal cortex, and in the other side, you see how FCD looks like. However, one of the biggest challenges of FCD is that in many cases, these lesions are difficult to detect by conventional MRI, which normally targets the microstructure and can reveal where the lesion is located in the brain. So surgical resection for these patients is not available. So that's why we're trying to figure out new ways to make these lesions more visible. So now to address this question, we first perform an experiment using an animal model of cortical dysplasia by injecting either saline solution or carmustine, also called VCNU, to pregnant rats. And then the resulting pups and the object of the study were 16 controls and 60 VCNU. And then we scanned them at 4 different time points. But for this presentation, I'm just going to show you the two far ends, like 30 and 150 postnatal days. So we acquired a diffusion-MRI acquisition where we use 3V values in 90 directions. And our last step of our experiment was our histological validation with immunofluorescence, where we target the neuronal nuclei and also the myelin protein for our cyto and myelo-architecture description. And then for our diffusion-weighted analysis, we follow the gold standard preprocessing steps, but we also use a Laplacian potential field to create our system of grid lines. So these grid lines have 50 columns and 10 equidistant points all across the cortical ribbon. So basically this system, what it's aiming is to sample our different diffusion maps as a common anatomical descriptor of the cortex. So here we use the DTI because wet spray use, but we all know that it has limitation for the description of complex architecture, such as the cortex. So for that, we use a multi-tensor based on a multi-resolution discrete search that can fit up to 3 independent tensors per voxel, but each one with its own eigenvalue and eigenvector. And because it is a per bundle description, it can disentangle into what is perpendicular or what is parallel in the cortex, so which is very convenient for this type of study. Then after sampling our different diffusion maps, we perform a longitudinal vertex-wise statistical analysis using a linear mixed effect model. And here again in the grid line system, just as a reference, the white circle is indicating the significant p-value. So for our DTI analysis, as we expected, we didn't see really changes at any of the metrics or either at 30 or 100 postnatal days. As I show you here in the bottom of the slide, I show you a single vertex of how the data is behaving. So in the x-axis, you see the postnatal days, and in the y-axis, you see the metrics, the values. However, when we look at the multi-tensor results, we did see some changes. So especially here at 30 postnatal days, we see how FA, parallel, and perpendicular are displaying these differences between group in what would be the deeper layers of the motor and somatosensory cortex. And less so, but still in a FA parallel, we see a small cortical area displaying these differences, but not really much in the other metrics. Again, here in the bottom, I showed you the predictors for a single vertex again. So the red asterisk is just pointing at these differences. But now, in order to understand what these diffusivity changes means in a biological context, well, we look at the stainings. We first look at the cyto architecture with the neural nuclei. And here in the control, the first thing we see is a very clear layering of the cortex. You can see here by colors, we see the 6 layers. And when we look at the BCNU, we see that there's a blur transition between the superficial layers 3 and 4, and also to the deeper layers 5 and 6. But now we look closer at that area, and these differences become a little bit more clear. So we see here again the boundary between the deeper layers, and we see that there's no really well-defined, and as well at the superficial. We see here 30 days and 150, and the yellow rectangles is the BCNU rats. But also we notice that the orientation of the radial neurons are not fully like an correct order either. So we think that this is probably one of the biological things that are driving our results for the multi-tensor. But we also think that the fiber myelin orientation is playing an important role here. So here, for our myeloarchitecture description, in green, you're seeing the MVP stain, so the myelin. And here at 30 postnatal days, I want to highlight with the magenta arrows how the BCNU rat is displaying a loss of the myelization process, along with some disorganization of the radial intercortical fibers in a dramatic way compared to the control one. But here, very interesting is that in 150 days, the myelization process did achieve the upper layers of the cortex, but we still see that the fibers are displaying a disarrangement of their orientations. But this is very subtle changes. So if you remember in our 150 days for the multi-tensor, we didn't see much changes. So we think that this is probably too subtle for that. And well, we're thinking that a different type of modeling to make this more sensitive, it's needed. And our last thing that we did, it was structured tensor analysis, where we compute coherency maps based on the local anisotropy, orientation, and intensity from the MVP images. Well, it's taken from the texture of the stains, and this was our results. So you see in the left-hand side, we see the control rat, and in the right-hand side, we see the BCNU. So 30 postnatal days, we see here with the yellow arrows are pointing at this large cortical area, displaying a lack of coherence. So basically here, a coherency in this type of maps, it means that there is or not a primary orientation of the fibers from the stain. So when we see here at 30 days, we see that this corroborates to what we've seen in the multi-tensor as well. And again, on 150 days, we see these slightly changes, but they're way more subtle compared to 30 days. But we still see that there is a lack or a loss of the coherence in the deeper layers of the cortex if we compare to the control one. And well, with this, I just would like to highlight a few conclusions for the study. And it's that we know that the cortex, it is a challenge for most diffusion-MRI applications, but we think that this patterning of cortical fibers and columns could be sensitive for different diffusion profiles. And the second is that this multi-tensor proved in this study to be sensitive for the identification and separation of these abnormalities, but we only see it basically in early stages of development. And for that, we think that for analysis, for the identification of these subtle changes in the cortex and this disorganization is needed. But we still think that it has a potential use for the detection of human FCD in the future. And there's actually something that we're trying now, so we will see. And while we know more, I would like to thank everyone for the Connectivity Lab and my supervisors, and would love to answer all your questions. Thank you very much.
All right, thanks, Paulina. Very interesting. Sajad, do we have any questions for her?
Yes, thank you very much. That was great. We have one question from Amy Howard.
Yes.
Beautiful work. The mouse cortex is relatively large, and you have high resolution MRI, meaning you can nicely detect these layer-wise differences. How do you think your diffusion MRI analysis would translate in human, given that the human cortex is several millimeter thick and diffusion MRI resolution is often limited?
Yeah, thank you. Thank you. It's an excellent question. Well, now that's a challenge for sure, because you have like 2-3 voxels, depending on the resolution, right, in the human cortex. But now we're trying with a resolution of 1.2 and to see if we can measure or if the multi-tensor can be more sensitive. But we still like we don't know yet because we're still playing with the data. But I think if you go with a lower resolution, you start like playing with SNRI, and also you can emit some data. So we don't know yet, but we're trying to figure out those things. But it is way more complicated compared to the rat brain, because, well, that's a large cortex and doesn't have the gyrus and all the complexity.
Thank you. I have a question.
Yeah.
Have you tried running or like fitting your data to any model to see if any of them work, or they are completely kind of their assumptions don't hold?
Yeah, not yet. Like this is something that we think about, but we first wanted to start with something like with a more with a representation instead of a model. But for sure, is the next step. One of my colleagues for the lab is using a B-tensor at the moment, so we start trying to move forward to using more advanced approaches for this type of malformation of this data. So we will see.
Okay, great, thank you. I think we can move on to the next talk.
Okay. All right. Thanks again, Paulina.